Today we shall see the major platforms used for target identification as well as target enrichment. We know that normally our target would be a protein, and I told you that protein is an unwanted protein which is formed out of mutation. So the primary goal of drug designing is to identify a new molecule which would inhibit the interactions of this unwanted protein molecule. Earlier I told you, if A and B are certain protein molecules required in the body for normal metabolic activities, and if a new molecule C is getting into the scenario out of mutation, and if C interacts with A and B, these are really unwanted interactions which will weaken the real wanted interaction between A and B. So under that condition, actually we can say the unwanted interaction is really the cause of disease. So the interaction between C and A or C and B, or both C and A and C and B. So under that condition, what we do is we will introduce a new molecule D, which would be later named as drug molecule, and the function of this molecule would be to set up selectively interaction with C, that is a unwanted molecule, so that this will inhibit the unwanted interaction between C and A and C and B. That is why I told you earlier, most of the drugs are inhibitors. Earlier we have seen the possibility of identifying potential molecules, lead molecules, starting molecules, template molecules, etc. So today we will see the technique to identify target molecules, and the first one among them is, normally you know that we will be using the ligand, we'll be using the gene databases, common gene databases, including the gene NCBI. So let us take that page first of all, NCBI. This is the primary database with almost all our biomolecules or all requirements, such as, so we have got the ClinVar, gene, genome, protein, and so on and so forth. So this would be our starting platform for generating identifying or generating the target. And under that condition, what we see is, so as for example, we can just give a query like cancer. If I put a query like cancer here, then all database means everything will be coming, everything, genes, then geo dataset, geo profile, publications, everything will come. At the same time, suppose your query is going to be limited to, say, gene alone, genes for cancer. So you are seeing the common genes which are therefore related to cancer. See that about 3284 genes are there. So automatically you can make your query again. So cancer, like pancreatic cancer, so instead of cancer we have made a query like a pancreatic cancer and see the genes or mutations responsible for them. Then we'll be getting a limited number. So at present we have got only 2683. But a major problem with this primary database is it includes genes connected to pancreatic cancer of all organisms. At the same time, if you make your search in a specific or secondary database where only what we need is only human mutations and human genes alone are going to be taken, then the best one among them is actually gene cards. So this is gene cards, and you can give the query any query within that. Then gene cards, mind you, gene cards is actually specific for human being, so no chance for any other organisms or gene of any other organisms to get into it. Moreover, you will be able to make a specific account in gene cards, like say I have made it with a username and password. You can make it. Now here if I give the query like a pancreatic cancer, pancreatic cancer, now what you see is about 12,558 results are there for pancreatic cancer. Then so all of them are directly or indirectly connected to it. So what I want to tell you is a large number of mutations or genes will be responsible for it, and in most cases what we find is a pancreatic cancer patient, if we observe the mutations happened or mutations which took place in that patient, what we find is more than thousands of mutations would be there together responsible for that condition. So it's very difficult to identify or pick up a single mutation and work towards a single mutation. Or if you think of identifying drugs aiming at or targeting a single mutation, that may not work out in most cases. And moreover, you see, if you want to see the major mutations going on, a direct method is you find the gene expression analysis, what you call otherwise as gene expression profile, and I will tell you how gene expression profile is going to be taken up later in the coming classes. So this is another method. Suppose you are having about 100 mutations or 1000 mutations. Here you come across 12,000 or more than that. So under that condition, suppose you want to check whether the mutation which you are going to pick up is relevant, because if the target is wrong, your entire process may be wrong, right? So whether you design the molecule or whether you modify a molecule or whether you start from a phytochemical, whatever may be, that if the target is wrong, your entire work is going to be a waste. So automatically what we would like to do is You make a primary screening based upon the importance or based upon the mechanisms or based upon the relevance, based upon the expressions. Because I will tell you a typical example. You know that BRCA1, here also we have seen BRCA1 and BRCA2, they are very, very common for most of the cancers, especially for breast cancer. BRCA1 is making a major role among females and BRCA2 is making a major role in males. But the problem is identification of BRCA1 or BRCA2 or expression of BRCA1 or BRCA2, if you make as a criterion for diagnosing breast cancer, in most cases you will get it diagnosed at the end, because BRCA1 and BRCA2 are very negligibly expressed in cancer samples. So if you take a sample of, breast cancer sample and check whether BRCA1 or BRCA2 is mutated, then the problem is due to the low expression of these genes, in most cases we won't be able to detect it. Otherwise you may have to make sometimes 30, 40 cyclic process in the RT-PCR. You know that in RT-PCR, so actually you are allowing that gene to grow. So this process you will be able to do several times, several cycles you may have to do. And in most cases what happens is, actually when you just see the BRCA1 or BRCA2, or when you follow BRCA1 or BRCA2 to test breast cancer, only in the final stage most probably you will be able to get it. Hence what I want to tell you is the expression profile is a major concern for us. In some cases, instead of BRCA1 and BRCA2, TP53 may be expressed, especially at the beginning itself. TP53 may be expressed. So under that condition, so what we can do is, so we'll be able to find the the profile, and based upon the profile we can pick up the genes in order. So sometimes you see that specific gene, instead of that specific gene we'll be able to start with TP53. You know that when TP53 is expressed, or when TP53 is overexpressed in a organism, that itself indicates that cancer is already there. Normally a definite pattern will be identified or observed for each type of cancer. So if the profile, the expression profile for breast cancer is known to us, of course BRCA1 and BRCA2 also will be there, which will be very negligibly expressed. At the same time, suppose that profile, that means that pattern is known to us, definitely we will be able to identify it early in advance. And this is what is normally followed during the diagnosis. So just like in COVID-19, you might have noted 2 or 3 genes were used for checking whether, or diagnosing whether the patient has got COVID or not. And it is presumed that these genes are well expressed. So normally well expressed genes would be preferably taken for diagnosing the disease. But at the same time, I have to say that for breast cancer, BRCA1 and BRCA2 are expressed. Hence, the first point which you have to remember is there will be thousands of mutations together going on for a patient. Even we may have to say that even before the disease is going to be diagnosed, these mutations will be there. And if the expression profile could be checked in the beginning itself, especially for the patients who are prone to the disease, suppose in a family somebody has got breast cancer, then definitely there is chance for other people from the same family to have it. So at least under these conditions, if you think of the profile, that means instead of checking BRCA1 and BRCA2, you may have to check TP53 or such genes which are having higher expression in the beginning itself, and see whether these genes are expressed early. And if they are overexpressed, definitely that indicates that that mutation, that process has been started. Then we can have a vigil on BRCA1, BRCA2, etc. Or you can start the treatment strategy in the beginning itself. So these are some of the ways which would help us to diagnose the disease early in advance. And mind you, there is a technique which we shall have it, especially the advanced session, which makes use of deep learning and image processing, deep image processing, what you call otherwise as, and based upon that, that pattern could be read very easily even from a biopsy image. So if the pattern is diagnosed in the beginning itself, and if we see these, at least these mutations or the expression of these mutations, then that itself would be a very early step to diagnose the disease. So with target, or if you analyze the target properly, or if you see the profile of targets which are expressed or overexpressed in the body, or if you identify the mutations which are there already present in a patient, definitely that would help us in predicting the disease early. You got it? So at present we have seen several mutations, and I told you BRCA, I told you the gene cards could be used for that purpose. And suppose you need to take all these 12,558 molecules, what you can do is you may just click on the export option here, and you can export the entire molecules, entire screen as an Excel sheet. So that is the possibility which you can do. Let me just check it. See, for exporting the result, what you need is you need to log in. So you can log in, then you will be able to export the entire result. Then you might have seen here the molecules are arranged according to the score value, and here the first one with a simple BRCA2, it has got maximum score. Then up to 503 pages spreading, so you could take the entire result as an Excel output. Then again you will be able to pick up a few of these molecules for your analysis depending upon your requirement. Like you can say those with score value less than 100, more than 100 alone will be taken. So in that manner, so this is one area where you can put up. So similar to the NCBI, you will be able to get results regarding the target or some idea regarding the target from it. And next possibility is what you can do is in here itself you will be knowing. So we have got, you might have seen gene, CARNA, MALA cards, path cards, VAR, elect, gene analytics, etc. So out of which CADD is going to be a suitable one for you, where you could give the disease, and based upon the disease you will be able to get the target. Like, so you can say pancreatic cancer, pancreatic cancer. Now what you see here is, for pancreatic cancer, so we have certain proteins, and you click on pancreatic cancer here. Again, you'll be able to see those mutations, or those actually it's an information with respect to the pancreatic cancer, and you could even see the major gene responsible for it, that is actually ICD11. Which has got scoring, we have seen that 250.116. So similar to what I want to tell you is similar to what you used earlier. So here for pancreatic cancer, we would like to see some mutation, some specific mutation regarding pancreatic cancer. So that is possible. Then similar to that, if you use gene card there itself, suppose I click on BRCA1 or BRCA2. So let me click on BRCA2 and I want to see the associated mutations. Now here what you see is, you will be able to see details of that BRCA2. So thereafter the external IDs, then summaries for BRCA2 gene, then genomics, then this actually the gene view or genomic view, and this is the exact gene, what you call as BRCA2, which is from chromosome 13, and its position is 13q 13.1. so this is that gene. Then again, if you go through it, you will be able to get the 3D proteins corresponding to that, major 3D proteins, highly expressed 3D proteins corresponding to this mutation. Now if you go to the bottom, again similar to whatever I told you, several results and information will be obtained. Here there is a part what you call as a string interaction network. So you click on it, we will be able to get the genes which are connected to BRCA2, associated with BRCA2, and definitely we will be having BRCA2 here, then BRCA1 is there, then FANCD2 is there, CDK2 is there, PLK1 is there, so on and so forth. So these genes together will be most probable genes, mostly associated with BRCA1 and BRCA2. Here also you will be able to export the results. You can export it. Then while exporting, you can take the tabular text output download, which you will be able to get in Excel. 1, 2, 3, 4, fourth one, tabular, short tabular text output format, means TSV format, TSV tab format. Then if you click here, immediately you will be able to get it in the form of .tsv, similar to .csv, which you can collect, which you can take in Excel. Got it? So this is another method, because if you know that okay, one mutation is already known to you, which is known to you as a very important mutation for the disease. So under that condition, gene card can be used for identifying other mutations which are associated with it. Then especially by using the string network, and if you download them, then definitely you will be able to get the mutations which are resembling or which are related to BRCA1 or BRCA2, and thereby the set of mutations which is relevant for the disease that could be identified. So that is one possibility for getting it. Then another method is we could use therapeutic target database. Therapeutic target database, that is db.idrblab.net/ttd. Normally this database is known as TTD database, therapeutic target database. Now here also you know that we could this is the home page. Then you can have the target sequence similarity, drug structure similarity, coronavirus specific search possibility is there, the target group, successful target, then clinical trial targets. Then that means even in the target, if you want to limit it to only successful target, means you can give the successful target alone. Then drug group, you'll be knowing the approved drugs, clinical trial drugs, pro drugs, etc. So if I click approved drugs, then I'll be able to get the approved drugs. For here I can give some disease like pancreatic cancer, or some mechanism like EGFR, some pathway. I'll be able to put some pathway like EGFR. Then the drugs targeting EGFR, all the drugs which are targeting EGFR, that will be shown to you. So similar to drug bank which we had earlier, because ligand part already we have seen, but here I just give you the therapeutic target database that will help you in identifying that also. Now similar to that, if a ligand is known to us, suppose you know a particular ligand like cetuximab, then TTD drug ID, then that drug information. So directly you could see for that particular drug about the drug, or instead of putting a disease, suppose you put a drug, from that also you will be able to get. Similarly, successful targets if you want to see, so there you can give the query. Then you will be able to give it the approved drugs, clinical trials, then patient data wise, suppose you need to know some trials also, or check it or evaluate your result with the trials or experiments which are already over. So that is also possible. So therapeutic target database is one such database which would add all these things. So mind you, everything is one and the same. Similar results you are able to get. Some additional correlation may be there. That is also, whether you start from NCBI, which is a primary database, or if you shift to the gene cards. The advantage of using gene cards is you will be able to specifically pick up the human organism, that organism specific genes or mutations, you will be able to identify. That is the advantage over it. Now I will tell you another platform, what you call as open target database. And here also you will be able to see find target for a given disease. So you click here. Now that disease you can give like pancreatic cancer, then there itself it asks us whether it is pancreatic neoplasm or malignant pancreatic neoplasm or pancreatic carcinoma. Now your search can be limited to it. Now what you see is you will be able to see the mutations here, important mutations here, That color code is also important, because you know that at present KRAS is going to be a major mutation with a score value of 0.81. TP53, that also has got 0.81. BRCA2, its score value is only 0.78. That means that score value based upon the expression, that also will be obtained. So similar to that, here if you see the somatic mutation, pathways and systems connected with it, pathway type also you will be able to select. So you can decide which pathway is going to be taken up by us, like metabolism related, because you know that at present cancer is treated as a metabolism disorder at present. So under that condition, you will be able to pick up pathway related only mechanism. I mean, what we have done is metabolism related alone. Then we can easily pick up a few mutations like POLT1 is actually a mechanism, means actually here metabolism related targets, then focusing on pancreatic cancer. So that is why pathway related, autophagy related 81, cell cycle related 296. So the pathways also, so you can, as a disease alone 950, without any specific pathway, if you just pick up the disease directly 950. Then gene expression 622. Here I put it up metabolism. Then in the metabolism itself, other possibilities are there. Then so similar to that, say whatever pathways are there connected to pancreatic neoplasm, 734 targets are there, so which would be classified into different pathways. That is the advantage here. So you can easily make a correlation with the pathway also. So earlier I told you, when the topics were introduced, I told you the importance of pathways. So definitely, so rather than just considering one target or one disease, a particular type of target, then you can specifically mention this mechanism is important to us, so that your drug will be focusing on that mechanism. Autophagy, as for example, autophagy related means your target should be, so if I click on autophagy, then let me remove metabolism. Then definitely you are seeing some different mutations, right? Like TUBB3, TUBB4B, etc. If autophagy alone is going to be taken for pancreatic cancer, mind you, so we have taken pancreatic cancer, that is a major search. So within that itself, pathway based mutations can be specifically identified. Hemostasis, then KRAS and all that. Here there is an option where you will be able to use gene expression alone, expression wise. So directly you can get into expression concept. And what you see here is KRAS is most expressed, mind you. So automatically for pancreatic cancer patients, sometimes say STK11, CDKN2A, etc. They are all specific mutations. At the same time, expression wise, KRAS is going to be having maximum expression value 0.81. So automatically, if you take a sample of pancreatic cancer, then so what we see is about 0.81% KRAS will be expressed. Then TP53 same amount, 0.81 value, so 81%, mind you, not 0.81%, 81% TP53 is going to be expressed. Then 80% SMAD4 is expressed. BRCA1 basically 0.74, that is his core value. So 74% BRCA1 is going to be expressed. That means actually if you just pick up a few of them at the end, you will be able to see that their expression is negligibly small, 0.47 score value. So 47% expression would be there. So if you make a simple real-time PCR analysis, most probably you won't be able to identify max, fan CC, etc. So what I suggest is, suppose you want to identify pancreatic cancer in the beginning itself, you may have to pick up KRAS, then TP53, SMA-D4, BRCA1, STK11. At least these 5 mutations may be taken up by you and see whether these mutations are there in a patient. Suppose these mutations are already reported in the sample, then definitely we may have to see that other mutations would be there which may not be that much expressed. Mind you, for each gene you may have to do the cyclic process, thermocyclic process separately, and you will be able to get the KRAS expression, TP53 expression, SMA-D4 expression, etc., which would be obtained easily as they are highly expressed in the sample, whereas remaining or last few of them that would not be easily obtained. So that is why if you put BRCA1, sometimes in some patient BRCA1 may not be expressed. At the same time, you see that KRAS is expressed, 80% expressed, and suppose TP53 is also expressed and SMA-D4 is also expressed, definitely there is chance for other mutations. So this is actually what is meant by the pattern and profile. Profiling of a sample, that is more important than identifying the mutations going on, right? And so with that, sometimes you may think like, okay, ligand targeting a single protein or a single gene or a single mutation, that is very, very difficult. So here comes the requirement for multi-targeted designing. You got it? A ligand that would be normally, that is expected to be also normally multi-targeted. So these targets you have to identify mainly, not a single target alone. Identify the major target. So suppose your interest is on a particular mechanism, then think of that mechanism. So you avoid gene expression. And suppose you want to check whether the gene is the mutation is DNA repair related mutation, then let me click here, DNA repair mutation, see the expression, and what I see is TP53 is going to be the most relevant one as far as DNA repair is concerned, then BRCA2, then PALB2. So if your design is focused on DNA repair alone, then definitely you have to pick up TP53, BRCA2, then PALB2, then POL, etc. as your targets. At the same time, suppose your mechanism is going to be digestion and absorption related mechanisms causing pancreatic cancer, then definitely you may click here. 10 such mutations are there which are going to be like GUCY2C, then SLC 54A, 5A1, then SLC 2A2, then GUCA 2A, then NPC 1L1, and so on. Then based upon your pathway of interest, cellular response, cell-cell communication, if you think that cell-cell communication related defect alone as your major target, then you can pick up here cell-cell communication. So read the mechanisms clearly and check or focus on a single mechanism or one or two mechanisms, then definitely you will be able to pick up the real target of your interest. Then so this is another possibility, what you call as open target platform. So far I told you about making a what you call as a simple search using NCBI, pick up all genes which are there within that 12,000, 15,000, 20,000, like that you will be getting, and you may not be able to work out with 10,000 or 20,000 mutations at a time. Then I told you, you could even use what you call as a modified platform, a secondary platform, which is used specifically for an organism. For a human being, as for example, gene cards could be used instead of NCBL. You will be able to use gene cards directly. So there also, suppose your interest is on a particular gene and correlated gene, I told you, we could set up, as for example, TP53 related, suppose you want. So what you can do is, you may click on TP53, then see the associated mutations with the TP53. Then again, suppose you need a simple search. So what I told you is, you may go to the here. This is actually interacting protein for TP53 gene. Click here, then that network will be set up with the TP53 as a central gene or mutation. So this together could be taken up by you. But suppose, I think in modern research you may have to use, see, the open target platform, where you will be able to pick up the pathway, everything you will be able to pick up. That is a advantage. So this is going to be a very relevant platform for us. And here also you may see that the I have picked up only pancreatic neoplasm. So instead of that, other possibilities are also there in this search. So pancreatic carcinoma, then also we will be able to get it. So once you get it in this manner, then you may click on, either you can click on the target classes. So these are the targets, different targets you can take, like enzyme based or something like that, or pathway based, or any of these you'll be able to pick up, like antibody. But for a designer, normally target class may be important to us. Like sometimes you will say ion channel related, membrane receptor related, then secreted protein related, structural protein related, surface antigen related, transporter related, so etc. You may have some interest, and normally you will be having pathway related interest for you. These are the pathway type. So pathway type, there, as I told you, each project may be, sometimes you see gene expression wise, immune system wise, metabolism wise. As I told you earlier, the present tendency is to treat cancer as a metabolism disorder, not a proliferation disorder. It will be treated as a metabolism disorder, and if it is treated as a metabolism disorder, slowly that will come as a lifestyle disease. And so here you can see programmed cell death, if that is going to be your concern. Then the sensory perception, so like that, different major pathways. So I think this would be a good start for you. You identify the target as well as pathways, and based upon the pathway and target, you pick up your most relevant target. So there also, I think what you may do is, you may pick up a few of these molecules. Then suppose you need to check whether a specific pathway based you need, like autophagy related. So click on autophagy. You are having about 80. So what I suggest is, out of which highly expressed genes like TUBB2, etc. So these genes, what I mean is, these few genes, TUBB2, from TUBB2 to TSC2 could be taken as the reference genes, reference mutations. Then if you think, you need to take 943 genes which are included for the disease, okay, that also could be taken. That means if you are considering that alone, then you will be picking up KRAS, SMAD4, etc. Several mutations will be there. So what I suggest you is, try to pick up few of the mutations which are most relevant for the disease, and a few of them, if possible a handful of them, 10 mutations, then whatever be there from your general search, you can have a general search, you can go through gene cards, you can go through the NCBL, you can go through any of the software, I mean, or you could even pick up few of them from references. So all these together try to make about 100 genes. So first set is you try to pick up about 10 genes which are mostly relevant to your specific project, like cell cycle based or something like that. I prefer you to use this open target platform, and from the open target platform definitely you will be able to pick up a few of the mutations. Then so you have got two sets of molecules. One is you have got some what I call them as the reference molecules, and they are going to be the training molecules later on. And the second one is we are going to have the entire query molecules or entire products which you obtain, output which you obtain right from the query. Then what you need to find is with this reference molecules, what you call as training molecules, you will check whether any other molecule is there from the remaining set, because you cannot just pick up a few molecules right from open target platform alone. So what we see is from open target platform you got about 10 or 14. Okay, take them as the reference or control molecule. Later on we will call them as the training molecule. Then whatever molecule which you come across other than this and which you think as maybe relevant to the disease, then all these could be picked up together as your second set. So two sets of molecules will be there. Now we'll be using a very broad, I may have to say very comprehensive, exhaustive platform. What do you call us? We will go to top gene, TOPPGENE, top gene. So this is a top gene. Opening. Now we have got top function, top gene, then top net, top gene net. Now first thing which you have to remember is top gene suite is a gene enrichment platform. Then so we will familiarize more with top gene. So actually top gene is going to be a platform covering most of the properties or most of the features related to biofunctionality. Like, so you have seen there A, first set is A, and that is actually towards making a training set. And for making the training set, what you need is just like what I told you earlier, you could think of diseases of your concern, like we picked up pancreatic cancer. Then you may think of the pathway connected with it. So you may pick up a particular pathway of your concern, like metabolism or something related to it. Then you may think of GO terms. GO terms means gene ontology terms, which we are going to see in detail here. Then suppose you need to think of some phenotypes like mammalian phenotype. So whatever could be considered for picking up the training set, that you may do. And based upon that training set is made, then Once you get the training set, again what you need to do is, you may think of C, whatever terms which are included in C. So we'll be using GO in detail, like molecular function, then biological process, cellular component. So these together known as GO. And of course one more term is there in GO, what you call as the mechanism or terms which are connected to all these together. At the same time, as far as the second part is concerned, we have seen here human phenotype, mouse phenotype, then so these are all different components which could be included for the learning process. Gene ontology could be used for the training process. Phenotypes could be taken or included for the training process. Mouse phenotype, you know that normally that in vivo would be done by using mouse as a model animal. So that is why mouse phenotype is also included here. Pathways could be included here. So these are all, whatever is included in C, these are all certain components which could be used for the learning process. So with that learning would be completed. So when you give the query, when you include molecules or when you include different mutations into the platform, and we will name these molecules at present as the training set, and training set makes use of certain parameters or certain features or certain fingerprints which are functionally relevant, mind you, otherwise what you call as a functionally relevant features which would contribute towards the function. Earlier I told you about descriptors. While considering solubility of sodium chloride in water, so in the same manner, biofunctionality depends upon these features. Which all features? As for example, gene ontology. So what we need is, we need to express the biofunctionality. What is that biofunctionality actually? That disease causing biofunctionality. This is actually it's a case of unwanted protein molecule, disease causing protein molecule. So automatically its major functionality is causing the disease, and whatever attributes are there in machine learning, we use the term attribute. So here also features, you can name them as biological features or fingerprints, and these features will be used for getting the training. Like actually training is a learning process. System learns by making use of these examples given by you as the most important examples concerned with the disease of your concern, pathways you picked up, geoterms you picked up, or whatever of your relevance you may pick up. Then based upon that, the features were identified within these molecules or within the sample set. Features were identified. Now you have come to D. In D, what you do is, these molecules or these features are going to be scored. That means actually a scoring scheme is set up, a ranking scheme is set up with these features, and you could say that if within the molecule a similarity score would be made. So the idea is actually, once the training is over, molecule or not molecule, that system will be able to know that property fully. If I am saying pancreatic cancer, so pancreatic cancer related target, what should be the nature of pancreatic cancer related targets? That even specific to this particular mutation, specific to this particular pathway, specific to this particular geotames. So that would be known to us. Thereafter, you think of B. B is going to be a major search where you may have hundreds of molecules. So you will call them as the test set. And in the test set also, what you do is you will make a mapping by making use of the protein-protein interaction possibilities, what you call otherwise as PPI, protein-protein interaction. Then based upon that, a scoring is made and you compare that with the training set. So the test set is going to be compared with the training set, and for the test set, a complete scoring would be made so that you will be able to pick up these molecules with high score along with the training set. Mind you, training set will not be analyzed in this manner. Suppose you need to check training set also later. What you may do is, whatever result which you are getting after the analysis within the test set, few of them may be used as a training set and do the process again. So you could minimize the training set. Suppose only 10 molecules are there, 2 molecules may be used for the training and the remaining 8 may be used for the testing, or whatever you want. If you want to know these properties, relevant properties for the training set, that also could be done. And this platform, this high-end platform is what is known as TopGene platform, and normally the researchers would use TopGene platform as it exhaustively screens or exhaustively finds the most relevant mutations, most relevant genes, functionally relevant genes to a large extent. So that is why this is normally the platform that is used. And now you have seen that platform TopGene, and as I told you earlier, whatever may be the mutation or whatever may be that, you may to start with, what I suggest is that open target you may use. Then you may think of a particular mechanism. Based upon that mechanism, whatever is the most relevant mutations, these mutations may be taken up as the training set. Then other mutations, whatever mutation is found in a literature or anything like that, any number also you can use, that could be taken as the test set. Now you may open TopGene. Now let me show you how that would look like. You click on here. This is actually the TopGene suite. Now here you have got different techniques to introduce your input. You can use SGNC symbol or enrich ID or ensymbol ID, uniprot ID or anything like that. Let me just give you a typical example here. Here, these are the training sets. Like only few molecules have been taken up in the training set. Then a very large number of set has been made as a test set. Very large. See, whatever may be that number, that doesn't have any problem. Now, in a machine learning approach, your first step will be identifying purity of the data. So sometimes what happens is, though I picked up some 10 molecules as the training gene set, some example may be irrelevant, what you call otherwise as it may be an odd man. So identify that oddness within that set. Similarly, for the test set also, if you randomly pick up few of the genes, definitely there also some unwanted alien mutations will be there. Alien means not fit to that. So this is the first thing which you have to do. What do you call as the gene enrichment, in the gene enrichment process, where the first enrichment will be to see the quality of data set which you have taken. So whether these are suitable to be included together. Just like sometimes the students who studied machine learning might be knowing principal component analysis or something like that. So I don't want to make any of these complexities. What you do is you may just pick up few molecules which you may think as very relevant. You may think I use it, and you may think that may not be relevant. If it is not relevant, immediately in this step that molecule will be kept out. You got it? So few molecules you may keep as a training gene set. Few other molecules may be kept as test gene set. Now you submit. Now what is going to happen here? The data purification or data cleaning is going to happen here. So let us see how it is going to look like. Submit, and it won't take much time. And what we have seen is training set, not even a single molecule is unfit. 7 out of 7. 7 molecules were given. All 7 molecules were included here because no molecule is found as unfit. Okay, right? Whereas for the test set, 158 molecules were included, out of which only 143 have been identified as relevant. So remaining molecules have been removed, or remaining molecules will be removed from the analysis. You got it? So this is what is known as the first phase of data purification or data cleaning. Unwanted data, if you collect from any unwanted source, that would be picked up here, it would be removed here, that would be dropped from the data set. During this process, it won't take much time for that. Then once it is over, now if you go through that, so here you see that 143 molecules are included, few molecules are ignored. These are the molecules ignored because of identifying that non-relevance. Now here also you could add, if anything, if you feel like okay, this I would like to see or something like that, then I would be able to find alternative for missing symbols. Sometimes what happens is that a symbol may be wrong. Sometimes if I put it like SCNTA2, now what I can do is I can just check whether such a mutation is there or not. So instead of that, if a new name is there, you include that new name and make your search again. So that also may be a reason behind ignoring a mutation. So most probably what you may have to do is, after identifying few of them as unfit, you can just make a search on it. Especially in NCBI, you will be able to make it. Now here itself you may put the query as gene. Now I want to check whether such a gene is there or not. CENTA2. What is written is CEN. A is there going on. 13 items are there, and here you can see that CENTA2, that new name is actually ADAP2, right? Here it is given. So instead of CENTA2, you may put CADAP2. See, that is actually the meaning. So the moment you see few molecules are unfit, that doesn't mean that you can just leave this molecule. Some days that molecule may be very, very important. Then here one molecule is a GATA4. It is in the training set. Training set and test set, same molecule you cannot put. So in that manner, few molecules have been removed. That we can understand. Now the second one, let us see, ADRBK1. So what you may do is you may check whether such a gene is there in preferably NCBI. Gene NCBI. The second example I am showing you here. Now here also, so what we see is ADRBK1. ADRBK1 is at present named as GRK2. ID is 156. So you may just make it as GRK2, not GDK2, GRK2 instead of ADRBK1. Now, so now what you can do is you may go to the learning page, go back. Now here you have seen ADRBK1, ADRBK1. So this you may remove. So instead of that, what you can do is GRK2. You can write it as GRK2. Now submit. I am just removing only one. Now let us see whether this has been taken up by it. Now 144 out of 158. GRK2, it has been added, right? GRK2, it has been added into it. So that is how you have to do. So don't leave this gene. What you may do is you may pick up names, because if you collect data from different sources, sometimes it may be data included years back and the same gene might be renamed. So that is why you check whether such a gene is there in NCBI, and if a new name is put up for it, give that new name and redo this. So this is the first thing which you have to do. Second one is you are seeing the training parameters. By training parameter, what we mean is features, and for features what you see is molecular function. So molecular function is going to be taken as a feature, mind you. Then biological process is going to be taken as another one. Cellular component. Now what I prefer is, let us pick up all. In the beginning, preferably you may pick up all. Then human phenotype, domain is relevant, pathway is relevant, puppet is going to be there, means you will be able to get the major papers where this particular thing is included, and diseases is there. So I think say once you pick up all, so thereafter other things you don't have to change much, like random sampling size, that is for coding genes, etc. Now let us start it. Start it. Now the machine learning process is going on with the data which have been included or which have been accepted and given to top gene. Machine learning approach is going on, mind you. So by the time that machine learning process is over, which all steps we did? First step is we have collected few most relevant molecules as what you call as the training set. Then any other molecule which you come across, these molecules have been included as the test set. Now you may do this machine learning approach and see whether any other molecule. Mind you, that doesn't mean that you can leave the training set. Training set is the most relevant one. Besides the training set, whether any other molecule is there, that alone you are seeing here, mind you. Suppose in this result as such, if you need the training set molecules also, what you may do is you can put the top molecules which are there obtained from the test set as training set and reverse that process. Then you will be able to include, or you will be able to generate properties of these. Now the first one you are seeing result, it is actually GO molecular function. Score is also given to you. Now you click here. Click here means actually the entire genes will be included here. Now I think, okay, my interest is DNA binding, transcription factor binding. Okay, DNA binding, transcription factor binding. Suppose I think it that manner. Somebody else may think it in this manner. My interest is only on transcription factor binding. Okay. So you are designing a drug molecule based upon the transcription factor binding process. Now what you can do is, suppose I am saying transcription factor binding. So what I see is here gene from input. You got such a column here. So genes from input. Click here. You will be able to get the genes which are there from the input, mind you, and where the genes are having this particular property. So if I think, okay, my interest is only on transcription factor binding, then I need to take only these genes. Which all genes? GATA4, then SRF, HAND2, then NKX25, then MEF2A, HAND1, then TBX5. Only these genes may be taken up by me. Now again I think, okay, I have taken only a few molecules, few mutations. I would like to see other genes which are having this particular property, transcription factor binding property. Now what I may do is, I may go to the last column where genes in annotation, means whatever genes are related to transcription factor binding in the gene database, all these genes will be given to you. You picked up only 7 genes during your search. At the same time, suppose your research is on transcription factor binding, you have to remember that 768 genes are there. So let me just show you here. These are the genes. Suppose you want to add any other gene from this data set. So this is going to be a data set, mind you. Here again, if you want, you will be able to prioritize and pick up a few molecules. And see what I want to tell you is, you won't be able to pick up all mutations corresponding to the entire mechanisms. You can pick up one mechanism, one specific mechanism you will be able to pick up. Based upon that, we will be able to design drug molecule. So here I have used only one feature, that is molecular function. Based upon that, I use the transcription factor binding. If somebody else is using some other, like with some from the same data set, what I mean is, we'll be able to make minimum, at present you have seen 32 projects can be put up. As for example, I will tell one student to see the chromatin DNA binding possibility. So designing a new drug molecule which is going to be useful for controlling the chromatin or focusing or targeting chromatin DNA binding process. Then I'll be able to get here only one. Here you see last one, only one gene is there. At the same time, suppose I need more genes, then I can pick up here. I'll be able to get 119 genes again. So that molecule will not be left out. That is the idea. So what I mean is, you might have seen papers in this manner, like the computational designing of new drugs from, targeting NFAT protein or SMAD binding protein or chromatin binding protein or histone deacetylase binding protein. So what I think is, if that data set is made by one student, then itself very easily we can pick up 32 projects, 32 different projects with the different targets, mind you. So you'll be able to get different targets and you could check. Suppose you already identified few ligands or few phytochemicals as useful for say the histone deacetylase binding, then we can see whether these genes are going to be suitable for some other targets also. Moreover, the histone deacetylate binding gene included here is for the specific disease of our concern. Mind you, same gene is going to be focused for other 135 genes are there, mind you. Same mechanism is going to be taken up for these 135 mutations or genes. Sometimes some other diseases also may be connected with this, that we'll be able to get it in this manner. So if you very sincerely, if you do the target screening, definitely from that I could give you about 30 projects. That is why in the beginning I told you
10 different projects will be given to you, then that time you might have wondered whether 10 projects would be that simple. Now itself you have seen 32 projects I could get from a single search. Suppose you search for pancreatic cancer. For pancreatic cancer itself, I will be able to get 32, minimum 32 projects, different projects, focusing on different targets. Then some other student may give the query like breast cancer. With respect to breast cancer also, similar projects could be made. So there is no limit for making the projects. They identify the target, different targets will be able to identify. This with respect to the first geoterm alone, molecular function. Now, if you go through biological process as the second, 45 terms are there. So 45 plus 5, about 50 terms are there. So here actually that process, biological process is taken into consideration. Now, as for example, suppose your interest is on cardiac muscle tissue development, then I will be focusing on these 7 mutations. These 7 mutations will be there. So especially for the cancer, for the heart attack patients, for developing the cardiac tissues, then these mutations or these genes will be taken as the major. So here actually process will be taken into consideration. Heart looping, cardiac chamber morphogenesis. So like that, anything and everything connected with that disease. In a primary process wise, biological process wise, you will be able to pick up a project. So if my interest is on muscle tissue development alone, then I have got 7 genes. Suppose I need to further work on other mutations connected with the muscle development, means I can pick up muscle tissue development. 662 mutations are there connected with this. So that is how you will be able to pick up a biological process with the targets. Plenty of targets are here. There itself, if you want to make a, what you call as a further screening or gene enrichment, same process can be carried out with these genes alone. With all these 662 genes, same process could be carried out. Then now we have taken the biological up to biological process alone. Here. This is muscle tissue development. Then this is actually the second feature, similar to that cellular component, that is a third geoterm. Now suppose your interest is on the chromatin based only. Now actually here only 6 such terms are there. Protein DNA complex related. So these are actually the cellular component connected with it. Then other things also, if you want, phenotype wise. So you are seeing the hypoplastic heart. Then suppose your interest is on domain, you could think of that domain like SRF-TF domain. Or designing new drugs working on SRF-TF domain. So you are actually using a domain. Based upon that domain you will be working. And you have got about 32 genes. Suppose SRF-TF gene is there, 2 genes are there from your collection, 2 genes are there. Then SRF and MEF2A are there. Suppose my interest is on further expanding it, then I have got 6 genes from the entire gene processes which are connected with this. So automatically I will be able to pick up these genes and perform. Now that is actually the idea behind using it. Then now pathways. These are the pathways selected. So 45 plus 5, 50 pathways are there. Now I can again make my query little more precise. Suppose I need the MAPK pathway, P38 MAPK pathway alone. Then what I can do is, actually only 2 genes are there which are having this pathway alone. So I can pick up them if I want, or if I want to find remaining 36, I'll be able to,
the 36 genes, I'll be able to take these 36, and with that, then suppose my interest is on adipogenesis, then definitely I can see the adipogenesis related mutations here, or I can see other 131 mutations. That means actually I'll be using a human pathway, a pathway related mechanism. EGF signaling pathway alone is of my interest. So these are the gene. This is the only gene connected with the EGF signaling pathway from my query. At the same time, 27 such genes are there. So suppose you pick all these 27, then that pathway alone can be taken up as your major thing. Like PDGF signaling pathway related, then you can, your problem will be designing new phytochemicals or potential phytochemicals focusing on the PDGFR beta pathway or PDGF signaling pathway. Then so in that manner, from pathway also you will be able to get plenty of examples. Suppose you need any reference material connected with that. So actually you are having this 45 plus 5, 50 reference materials are there directly. You don't need to make further queries or anything like that. From here itself you will be able to see the reference materials, and it goes on in that manner, co-expression. Now basically for a designer, our major interest will be to pick up molecules based upon geotimes pathway. These are the major terms which we are going to take. Geotimes plus pathways. Now suppose you want to see the molecules, genes which you included along with the gene ID, and each term you are getting in this manner, biological process. Then this is actually score wise put up. If I click here, now I am getting them according to decreasing order of score value with respect to geobiological process alone. Now if I click here, I am getting them according to molecular function alone. Means, so once I pick up molecules, then again a similar process I don't have to do. Right from the scoring, I'll be able to pick up the right molecule. So suppose you are having some 10 molecule with respect to a geo process or something like that, then if you want to again score them, then directly from this total or cumulative result we'll be able to get the result. Then so this is the top gene. So at present I told you about using top gene, and I think by this time you might have seen the possibility of increasing your project, then expanding your project. Then so what you may do is you may just start with some disease like a pancreatic cancer or breast cancer or ovarian cancer, if your interest is on cancer. Otherwise fever, some fever, COVID-19 or whatever may be, that you can just give that query. With respect to that query, whatever genes are there, genes are obtained, these mutations may be picked up, and as I told you, using the open target, you may pick up the most relevant one with respect to a particular mechanism if possible. Then your work also will be with respect to that mechanism, with respect to that pathway. So here itself you will be able to make a very comprehensive screening right from open target itself. Suppose your query, you feel like okay, it's very inadequate and you are having some more mutations with you, then definitely you may go through top gene. With the top gene you may pick up the other relevant genes also. Then there you'll be able to see the pathways within them directly without using any other tool. The pathways, then the mechanisms, then the geotimes, etc. So based upon that, we can very easily pick up the mutations, genes, or the pathway at least is very relevant to us, because based upon that pathway, whatever mutations are there in the gene card or in NCABL, then all these will be obtained directly from the top gene results. So in that manner, you pick up the target. Then, so at the end of this project or end of this experiment, you are expected to pick up at least 10 to 15 mutations. 10 to 15, what I mean is 10 to 15 mutations. Then write their properties, like whatever property is there for these protein molecules. By property, what I mean is mechanism. Suppose you know the mechanism. These are the mechanisms given by these, or these are the pathways connected with it. So like that, whatever information is there with respect to each gene, that also you may write. And here, so we have used a machine learning approach, mind you, and in the machine learning approach, we have made it, I mean, as comprehensive as possible by adding the possibility of interaction, also biomolecular networking. To some extent, we have included in the screening process, still to ensure that biomolecular networking is completely included in it. In the next class, we shall see biomolecular networking in detail, and whatever molecules which have been picked up by you in this manner, these molecules possible interaction would be picked up. Reason I will tell you, no biological system is 100% predictable. That is known to you because of the smartness enjoyed by biomolecules. Whatever may be the prediction technique. Here actually you are using a fuzzy logic prediction technique algorithm. Whatever may be that, there is always an element of uncertainty. A black box is already there. So just to come out of it, what we do is we will see the possibility of interaction among these protein molecules or among these genes. Then actually if I write BRCA1, it stands for a mutation, it stands for the gene, it stands for a class of protein, mind you. So BRCA1 protein. Then within BRCA1 protein, several 3D proteins will be there, mind you. So whenever I use the term BRCA1, actually what I mean is, in the beginning, if I introduce mutation to you, BRCA1 stands for mutation. Slowly I'll be turning to the genes, then BRCA1 stands for the genes, the particular gene. Then slowly if I come to protein-protein interaction networking, BRCA1 stands for class of protein, mind you. The interacting molecule is protein, not gene. So actually protein-protein interaction, which we are going to see in detail in the next class. So the reason behind following protein-protein interaction networking is, you know that all biological functions will be as a consequence of molecular interaction. Actually, molecular interaction is much more realistic. So we'll be able to pick up and characterize molecular interaction to a large extent. At the same time, functionality is very, very uncertain because of the smartness enjoyed by biomolecule. That is why by machine learning approach alone, QSAR analysis, what do you call us? Qualitative, structure, activity, relationship. So all these are not very relevant, very acceptable to biological systems, mind you. So that is why QSAR is not very important in biology, whereas it is important in chemistry. In chemoinformatics, QSAR is going to be very important. But if you apply that to biological molecules, protein molecules, etc., that may not work out because of the smartness enjoyed by biomolecules. So into that, we should have to right from functional domain, we may have to come down to a molecular domain. So that is why from functional domain, you might have got few mutations are relevant. Now we have to see whether this same correlation is going to be there, same result is going to be there within the molecular domain. So that is why we are doing biomolecular networking and confirm that these targets are very, very relevant for your project. Then only we will think of 3D proteins followed by docking. Okay, right? I think today's discussion will help you in picking up the right target for you. and you may pick up, as I told you earlier, at least 10 mutations of relevance for your problem, any problem which you can have for your experiment by today's experimental session. Then by today night this video will be uploaded in the channel. So you may go through the channel every now and then to see the updates, and I don't have to tell you that it is uploaded, because normally, mind you, it will be uploaded in 1 or 2 days. It will be uploaded. If possible, we shall see that this could be, if possible, we will do that by today evening or today night. Late hours, this would be uploaded. Okay, right? I think it's all right. Hello. That is enough for today.